Meiosis occurs in sexual reproduction to create sex cells with half the number of the chromosomes of the original cell. It's consisting of two cell divisions, meiosis I and meiosis II. Meiosis I has 5 phases: prophase I, metaphase I, anaphase I, telophase I, and cytokinesis. Interphase takes place prior to prophase I.
Prophase I of meiosis has 5 substages. These substages are leptonema, zygonema, pachynemia, diplonema, and diakinesis. Leptonema is characterized by partial condensation of chromatin leading to the creation of chromomeres that represent points of differential condensation. Zygonema is characterized by the appearance of bivalence, which represent 2 thicker, more condensed homologous chromosomes. Pachynemia, the synapse-terminal complex is formed and will now facilitate crossing over. In diplonema, the recombined non-sister chromatids of each homolog pair separate, slightly revealing X-like crossed areas or chiasma. And diakinesis, the chiasma terminalizes towards the telomeres. Metaphase I. In metaphase I, tetrads are aligned along the 3D equatorial plane or midline plane of a sphere-like cell. Centrioles or centrosomes are exactly 180 degrees on opposite sides of the cell. Anaphase I. During anaphase I, the tetrads separate, the bivalents separate, the paired homologs separate, and the paired homologous chromosomes separate. All 4 are ways of saying the same thing. After separation, the daughter chromosomes, which have 2 chromatids each, migrate to opposite poles. Telophase I. The chromosomes become less tightly coiled and less apparent, or we say the chromosomes decondense back to chromatin. The nucleoli reappear and the spindle breaks down. Cytokinesis. This is the division or pinching of the parent cell's cytoplasm to form 2 separate daughter cells in animal cells, or the formation of a cell plate from Golgi-derived vesicles in plant cells.
Meiosis II also have 5 phases: prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis.
Prophase 2. During prophase 2, a haploid set of chromosomes, each a pair of identical chromatids joined at the centromere, thicken as they undergo condensation and become tightly coiled within the nucleus. A mitotic spindle starts to form within the cell, outside the nucleus. The microtubules lengthen and the centriole pairs begin to move towards opposite ends of the cell. Metaphase 2. During metaphase 2, a haploid set of chromosomes line up in the center of the cell along an equatorial plane called the metaphase plate. Each sister chromatid is really no longer a true sister due to crossing over in pachyderma prophase 1. Anaphase 2. Anaphase 2 begins as the microtubules on both sides of the chromosome pair pull on the attached chromatids. This pull separates the related but now different sister chromatids at the centromere and begins the movement of the chromatids to opposite ends of the cell. Telophase 2. The cell elongates even more during telophase 2. A nuclear membrane begins to form from fragments of the parent cell's nuclear membrane around each set of chromosomes at the poles of the cell. The chromosomes become less tightly coiled and less apparent, or we say the chromosomes decondense back to chromatin. Cytokinesis 2. This is the final step of meiosis 2 and is the division or pinching of the parent cell's cytoplasm to form 4 separate daughter cells or gametes in animal cells, or the formation for spores via a cell plate synthesis even in plant cells. The cells of meiosis are terminal for animalia. They will not undergo any more divisions, but in plants, the haploid spores can divide in a cycle that uses mitosis and generates haploid daughter cells of a multicellular gametophyte generation.